Good morning. The title of our work is Color Preference Flexibility of Wild Butterflies in two co-occurring mock verbains under different pollination contexts, and is part of the doctoral project that Eugenia Drewniak is undertaking at the Universidad Nacional de Córdoba in Argentina. The feeding behavior of pollinators can have direct consequences for plant reproduction. Some species show strong innate color preferences, such as the Japanese swallowtail that only visits the yellow or red circles when these four colors are offered. Besides, some butterfly species show strong floral constancy, restricting their visit to a single flower color. In nature, several species show flexible preferences. For example, in a previous experimental work, we observed that naive individuals of the passion butterflies showed no preference for red or purple flowers. However, they were able to discriminate these colors as different, and they fastly learned to associate any of the two colors with a sugar reward. The goal of our work was to study the feeding behavior of the butterfly assemblage that visits two co-occurring mock verbains and their natural hybrid under two different pollination contexts. The study was carried out in a natural community and in experimental plots. In these two pollination contexts, we calculated the visitation rate of each butterfly species. We analyzed the interaction between the mock verbains and their pollinator assemblage using null models. We recorded the morphological trait matching among flowers and pollinators. We measured nectar reward. Finally, in the experimental plots, we evaluated the floral constancy of butterfly species by recording the visitation sequences of individual butterflies. Here you can see the pollination networks. We recorded the pollinator assemblage of 18 butterfly species visiting the red, purple, and pink flowers in the experimental plots, and 10 butterfly species visiting the red and purple flowers in the natural community. The hybrid wasn't included in the natural community network because we didn't observe any pollinators visiting the pink flowers here. Only 2 hybrid plants with few flowers were recorded in the study area. The width of the black bars show butterfly and mock verbain abundances. The width of the lines connecting a butterfly and a mock verbain show the intensity of the visitation rate. Colors of these lines represent whether the observed pattern differed from a random expectation taking into account species abundances. Orange shows higher visitation rates, light blue lower, and white no different from a random expectation. For example, species number 1 visited the red flowers in a higher proportion than expected. In general, observed trends were mostly congruent between the two pollination contexts. In this graph, the black bars show the proboscis length of the butterflies, and the dotted line shows the correlative length of red, pink, and purple flowers. Those species of butterflies that visited the purple flowers in a proportion higher than expected by chance, like species number 4 and number 7, had proboscis shorter than the correlatives of the red flowers. We found that red flowers offer significantly more sugar than purple and pink flowers in the two pollination contexts. And finally, in regard to the floral constancy, most species show a strong to moderate constancy, with 3 species being mainly constant to red flowers and 3 species to the purple ones. The 3 species that had consistent color preferences to purple flowers had proboscis shorter than the correlative of the red flowers. Similar results were reported by Briggs for other skipper species. Interestingly, skippers do not have visual receptors for red wavelengths. It is possible that the exclusive visits to the red flowers are related to the higher amount of sugar. These three species show a flexible feeding behavior.